my screen. Okay. Okay, I forgot to change the date. It's actually June 1st. So our overview, we're just doing announcements, updates, and then FRR, like how it went, any details about that. And then the upcoming for announcements is pretty simple. It's just competition. Ali's the only one in this meeting. So Ali, you know, it's the week of the 20th. Hope you're ready. Glad to have you back. It will be live streamed, so I'm sure we'll send the link to that 2.5 weeks from now. So it's big crunch time right now. And then this was just like a congratulations and good luck on Ali's or Drew's internship. Sorry, Ali. He's actually leaving today, which might be why he's not in the meeting. But he's leaving today to Ohio for his Amazon internship. So I just wanted him to know that we appreciate his work and we miss him. So updates. This is just a recap for what happened in lab last week. I sent a payload message about it already, but I just wanted to like update further, which is our newly borrowed GoPro. We tested it out and Frankie tested it out, and it allows video preview, which is really good because now we can focus like the camera and make sure the lenses can see stuff. And then it can record up to 4 hours. It still gets hot, but there's way less problems. And it seems to be in really good condition compared to the last one. We're still deciding on a time lapse versus video. I really would like to run one today just because I know the time lapse will keep it from overheating faster, and it'll also like save memory. And then yeah, 60mm glass Petri dishes instead of 70mm. I know I messaged you, Ali, about like making a new mount for it. Have you been able to like look at the old mounts at all on CAD?
Yeah, so I tried to make a new mount for it. The difference is that for the 70-inch one, there was like a base and then a lid that slid on top. But for the 61 that you sent me, is a base and then the lid actually covers the whole thing. So I just made a simple like cylinder hold for it.
Okay, cool.
And that should be on the Discord already.
Okay. And then yeah, just added risk in case of broken glass, you know, that's like the thing. And then we'll get into that with going over the FRR. The ballast, so it won't be an additional heat sink, is what was decided with Irvin. We just need an approximation of weight needed. I feel like we can reuse this ballast. We could go with a lighter ballast because the Petri dish and some like the power bank will be heavier this time. That'll be like less of a worry. And then our lens mounts are practically complete. But I typed this last week before the FRR. So we will have to work on the mounts. And then the power bank has been purchased. I feel like I brought it. It would be really bad if I left it in San Antonio. So I'm fingers crossed I brought it, but if not, we do have it now. And I can run tests at home if it is there. The headlamp was purchased, it's in the lab. One thing we're working on is like positioning it and making sure that it's the correct brightness, because if it's too bright, we won't be able to see it at all with the GoPro. So what Drew and I did was layered like tape over it to kind of like compress the light. And then we angled it a certain way so that you could see through the lenses at like a practical aperture. And then yeah, more rubber washers and grommets will be purchased for more stabilization. But I also think adding the ABS to the mounts will help a lot. And then regarding instant packs, I'm going to Hobby Lobby to purchase the plastic that we need. And then we can get started on that whenever. We do need proper PPE just because ammonium nitrate is like a skin irritant and all that stuff. So we'll probably do some work outside or in like a fume hood.
Yep.
Any questions? Sorry.
It's all good. Yeah.
Cool. So the FR recap, You weren't here, Ali, for the presentation, right?
I missed most of it. Yeah.
So the recording is there. I'll go into like the FRR. Oh, I actually have the FRR on there. I'll go down to like payloads portion. It's a good read, you know, just to have like all that information like known because they will ask us a lot of questions. But regarding payloads, I went over payloads purpose, which I feel like I haven't stressed enough with the payload team, like what we're actually doing. So that's like in depth. I mean, I feel like we all know like the big picture of it, but just seeing how like pressures and accelerative forces of spaceflight affect the reproduction and growth of E. coli. One thing that Randy nitpicked was pressures and accelerative forces. Um, I feel like we decided on accelerative forces definitely after the Q&A, just because he stressed like the 4Gs of force that are going to be on it, and acceleration will be monitored by the avionics bay, so we can cross that and make a graph. So what happens is we're asking the questions, or our alternative hypothesis is our assumption to the question. It's like what we think is going to happen. And then our null hypothesis, which is HO, is like what's predicted already by like nature without us like asking this question. So
Okay.
then we would apply our results to studying infectious behaviors and like gastrointestinal health. And yeah, that's pretty much it. Our growth rate that I calculated, in short, that's pretty much A is like the initial amount of E. coli, and then it grows, so it doubles, which is where the 2 comes from, and then it's to T over 20, which is like the amount of minutes divided by 20 because it doubles every 20 minutes on average, like in ideal circumstances. If we want to convert it to seconds, we can just convert the 20 to seconds and then do that instead, you know. But that's just like our basic growth curve. The less than, like the y is less than, is pretty much just us saying that the growth rate will decrease. It's just our alternative hypothesis going with our question. And then, yeah, so I just explained like why we're using E. coli. So the human gut microbiome is like a reservoir for commensal E. coli, and then UPEC, which is like the uropathogenic one. The uropathogenic one, how E. coli works is when strains like interact with each other, they can become pathogenic, which is the ones that cause infections. So studying E. coli like overall will tell us like if humans like get an infection with those 4Gs of force, we can determine how fast that infection will like spread and like the incubation period of that. But also studying commensal E. coli, which doesn't affect us at all, like it's not infectious, and it's actually like good for us. Commensal E. coli being in our gut microbiome pretty much takes up the resources and like all the food that they would feed off of, so that the new like other bacteria that want to come in don't have anything to feed off of, so they would die. Like it pretty much kills off the other ones because of like natural selection and stuff like that, because they're more fit in that reservoir. So that would, I guess, talk about like the overall gut health of astronauts. I just talked about like the reproductive phases. So like the lag phase is like, I would say we could make our culture either like before we ride there or like right when we get there. And that would give us a good like 1 to 2 hours before it begins to like reproduce at that like ideal rate. But then we have to like act within that rate of recording it, because then our stationary phase kind of like levels it out. Um, and then I just talked about like what I've told y'all before, which is it's not oxygen dependent and it's temperature resistant. So we can really just focus on how like the forces affect how it reproduces. And so another thing that I kind of updated recently is instead of dyeing it on like normal agar, which is I'm gonna test like the dye on normal agar versus the McConkey nutrient agar plate. The McConkey agar is like a lactose fermentation agar. So it's like originally pretty red, And what it does is like E. coli will reproduce on it. And because it ferments lactose, because it's gram negative, it shows up as red. And any other bacteria that like could contaminate it that's like gram positive will show up in a different color. So it isolates the gram negative bacteria, and it pretty much makes it easier for us to see. But also, if we do have contamination, our risk of like mistaking that for E. coli will won't really be there as much because it'll be in red, like the E. coli will be isolated. So I feel like that would kind of make our observations easier. So that's why I decided on McConkey agar. And then, yeah, our 60 millimeter sterile glass petri dish. And then I'll get into like what they asked about that and everything. Yeah, and then I went over like the camera magnification. I feel like we've been over this so many times, so I won't go into it too much. And then I haven't really like showed y'all the math. I kind of just gave y'all figures and was like, if you want to work on it, you can. But I did go in depth about the math just with how we place the light source, because Drew and I were kind of on like crunch time last week, making sure that you could actually see it. And we got really hyped when you could see it, because that's pretty tough to do with like an efficient amount of space. So what I did was I angled the headlamp at an angle to where alpha equals 30 degrees. So like this angle right here. And then so then we looked at like the numerical aperture formula. So N is the refractive index. I looked up the refractive index of McConkey agar. I couldn't find it. How you do find it though is you have to take the salts and the sucroses, like the salts and sugars from the agar, and percent it, like make a percentage of that versus water. So like proportion that. And once you find a percentage of that, there's like a graph where like this percentage equals like this refractive index for like your medium, because every medium has like a different refractive index. So like it says on there, like air has N equals 1. So N equals 1.335 for our specific agar, which is what I plugged into there. And then sine of 30 degrees is easy enough, just 1/2. So that's how that became the number we got. And then the diameter of the bottom lens is conveniently 25 millimeters. So I plugged that in, which is like the diameter divided by 2, to get F, which is like that focal point. So the focal point of the bottom lens, which is the 40 times lens, was then 18.73 millimeters. We can double check that math in the lab because I did kind of rush it and like try to double check it, but I couldn't really. And then there's these focal point like models that I use. This is on like a 1-to-1 magnification, so it's pretty proportional. So then what you do is like, this was a 1-to-4, so this was just that back down into millimeters into like this specific one. So then it's 4.68. So what it was is like, I tested it and the spacers we have are pretty close. So when we did assemble it in lab last week, it was extremely visible on like a 400 times plane, because we were actually like pretty accurate to what it was already. What we can do is just like 3D print like the spacers again and just cut them down at those lengths and double check everything. I went over the structural components. I'm not going to go over that again because I did do that in the payload meeting last week. And I felt like I went more in depth on those slides for the payload meeting than for the FRR slides, just because like I don't want to make the FRR too long for them. And then we just talked about like PLA, which I can get into with the Q&A. The cooling system, I just talked about how we're going to fit the volumes. I have the base proportions of the like reaction. Another thing is like we will be able to tweak the amounts of everything so that the reaction stays colder for longer and like everything like that. So that's just the research that's going to go into the cooling system. And then I'll get the plastic and everything, because Drew measured the plastic last week and I found plastic that was a little thicker than that, but I also feel like we could work with it pretty well.
Right.
And then just expected results. It's inferred by us, like when we were asking these questions, that it will be affected, especially based off of our sources of like last studies that I compiled. There's already like experiments where they put E. coli in like pressure chambers, and those were changed off of like certain like skews of data. And then yeah, I just talked about how our recorded footage is going to be compared against what's measured in the like by the avionics. And then I feel like how we would calculate it is we'll take the surface area of like the cell growth calculated across each frame, and we can measure it by like square millimeter. And then what would happen is when we do get those numbers, we'll have our expected values, which we can calculate just off of the curve, like the base curve that we have. And then we have our observed values, which is what we observed in the footage. And when you put those together and you map that, you do a chi-squared goodness of fit test, I can type it into the code like pretty fast, and it'll give us a p-value. Our critical value of 0.05 is just the like, I don't know how to put this, but it's like the most reasonable critical value to have in biological statistics. Every experiment I did, like even with genetics last year or last in the fall, we put 0.05. It's just like a base number. So if our p-value is less than that, I think we reject our null hypothesis, meaning that we can infer it leads us to believing that our results are correct and it did affect like the reproductive behaviors. If it's over 0.05, we'll conclude that our experiment was inconclusive.
Gotcha. Okay.
Yeah, so that just it pretty much just checks our works and tells us like, did you have a good experiment? Like this is like the accuracy of like the information you received. So I think that'll help us in like analyzing the results and being valid on whatever we get. Our risk matrix, I kind of just like copy and pasted what was on there I just took a screenshot and put it on there. They had a lot to say about the risk matrix,
Yeah.
which we can go over because it is, I mean, like our next thing for the meeting is like, how did it go? Which it went fairly well. It didn't go terrible. I guess terrible would be that they were asking a bunch of questions and I was like, oh, sorry, I don't have an answer. So I did in fact have answers, thankfully, and Gustavo prepped me like behind the scenes and was like, what are you gonna do with this? How are you gonna answer this? So I'll get into that. A lot of pressure was on Payload, like you said, like before I started recording. They'll kind of nitpick like one thing and focus on that thing the whole meeting, which we happen to be that one thing. Yeah, so Q&A recap. Most of the stuff was about the risks. Randy was like, why is the charging cable falling out of the plug? What is the mitigation for that? The charging cable is not falling out of the plug. However, he did stress that that is like something that happens way too often at launches. So it is like a valid risk that we put on there. So it's not bad that we put it on there. He was just wondering why it would be improbable. And he was kind of upset that we put it at improbable because it's pretty occasional, it's pretty likely. One thing I did tell him, because he wanted a mitigation for that and how we would bring it to improbable, we're gonna assemble a charging lock. I feel like we can just do it out of zip ties.
Yeah.
So we'll just zip tie like around the power bank and then zip tie around the cord and then zip tie those together and that'll like lock the cord in. We also learned about risk matrices and how to properly do them. I guess it's like we should show the process more of like taking all this stuff and having it start up here, like in the red, and then going through our process of mitigations and how we brought it down to like yellow and green. Another thing they asked about was bacterial contamination. I was extremely ready for that just because you're allergic to penicillin, Ollie. So we definitely shouldn't have bacterial contamination in general, but definitely not now. So I talked about like using proper PPE, we'll use gloves, we'll use masks, um, we'll decontaminate, like we'll sanitize, and just like everything like that. We'll also use like lab coats. I mean, we have proper PPE for like all of the other stuff, like the cooling system that we're using and like making the agar and stuff. Another thing we can do is spray everything with microban, which kills bacteria for up to 24 hours. So we'll spray the whole bay, we'll spray all of our equipment, and we should be good. And then the petri dish itself is glass, so it's airtight when it shuts. And then I said we'll take clean wrap, which like pretty much seals everything in, and we'll cover that in microscope tape and shut that even more. And the microscope tape is like super porous, so anything that's like the size of bacteria gets stuck in the microscope tape. We won't have to worry about that. And even if we do, the commensal strain that we're using is not infectious. I did a lot of research on the strain. It will not cause infection, but still, it's not a good look that like our petri dish like flew open and we got bacteria everywhere. So that's that. And the petri dish is also like highly chemically stable, so there's nothing like seeping out or anything like that. They had other questions regarding a lot. I feel like I'm gonna rewatch the FRR and write notes on everything else that they said. Most of their questions, however, were about our mitigations, our bacterial contamination, and then our 3D filament. The big stress was on the 3D filament. And then Alex, who we're using his GoPro, so like he definitely knows. But he says like there is like a risk with like keeping the GoPro plugged in and that port open. So we'll figure out how to mitigate that. But he just wanted to know, like, did we run our tests already? Did we test how it worked and stuff like that, which we did last week. It was something that we really needed to do before the FRI just because we just got that new GoPro. But yeah, I will go over like rewatch the video and then write it down on the notes. I have a doc for like post FRR notes. Did you have a question? I'm sorry.
I was just thinking, I think for the risk matrix especially, they just wanted to see our process. Like they wanted to see how we mitigated a lot of that stuff, and we just didn't tell them. So
Yeah.
I know like when we have to actually present again for I think the MRR, we can just make sure to include that part, and that should probably be most of it.
I hope so. You're right. I think just including that thought process and like, yeah, how we got it from here to here is what they really stressed on. I think the entire team, like every sub team wasn't really doing our risk matrices correctly. Like we weren't really doing that correctly. And that's what the other leads were telling me. So I was like, at least Payload wasn't the only one who did it, but our risk matrix was a little more under attack, just because they've never even seen Payload before for this year. So like at the PDR, we would have been able to show them like, oh, these are our original risks. And then we would have gone through that process of mitigating that. I just have a doc for like post FRR notes. So anyone that has like anything to say, any questions, if you like watch the recording, oh, I got 10 minutes, sorry. If you like watch the recording, just write it down in those notes. And then I'm probably gonna host another meeting like tomorrow or something to discuss after I rewatch the FRR, just because I haven't had time to do that. Yeah, I just within this next 10 minutes, I want to talk about the filaments because one thing we didn't do when we were testing is we didn't test it under like
Below.
Yeah, we didn't test it under a load, so like the tensile pressure was not there. We didn't put like any weight on it under the heat. So even Randy stressed that like PLA by itself will be good under those temperatures, but under 4 Gs and the heat, it may not. So the alumni really wants ABS. They really stress ABS. I did bring up that like you and Kevin did really good on the trade study, and you did say like ABS is a pretty good shot for us. But I think we went with PLA just because of the availability and then like the prices. But the alumni did offer to give us some ABS, and then I'm sure we have money in the budget now for ABS. So it's just converting those to ABS or like incorporating ABS in that. Do you think that's possible?
Honestly, I don't have a way to print it. If Gus or someone else is able to print it for us, I don't really see a problem in using ABS.
Okay.
We just have to get them printed as soon as possible, and so we can retest them and try to make sure they fit. But yeah,
Yeah. How has like the CADs been?
I'm not 100% sure on the new cat that I did when they told me to change it during the Q&A part, but I just want to test that. But other than that, I think they should be fine. We
Okay.
can always like cut them and make sure they fit better.
Yeah. Do you wanna I'll look over that today with you in lab then.
Okay.
Okay.
Do you know if someone's able to print with ABS though? Because I'm not sure if you can.
I hope so. Um Nobody said like I wasn't told. I feel like if we couldn't print with ABS, Gustavo would like say it during the Q&A that we couldn't print with ABS.
I know with most 3D printers, they usually are able to do a couple different filaments, and usually the two common ones are PLA and ABS.
Okay.
So I would assume so, but
Yeah.
yeah.
I think I also feel like if the alumni is offering to give us like a couple rolls of ABS, that if we really needed it, we could ask like Javier to print it. And just like give them some of the budget for it. But yeah, and then another thing I want to put on the FRR notes, the post FRR notes, is our takeaways, like what we learned, what we struggled with. It's going to be kind of like the personal statements from like the progress report. It's going to be like what did we learn, where did we like, how was the journey, and then also like any pointers and anything like maybe for this basis of this we should run an FEA on payload with analysis, or we should talk to structures more next year, or we should do this. Those are just like the takeaways. At the end of the FRR, they had their takeaways. I think would be really good for Payload to have our takeaways. So yeah, I think, I mean, you and I will have a lot more time in lab to like discuss everything. I can also record some of that for the team and just post it. But overall, that was pretty much all the meeting. It's just upcoming. We do need to go through the pre-flight checklist. We have a lot more to build off of it because our pre-flight checklist from last year for Oberon is really, really short and there's not much information on it. So just having that, and then we already have the hands-on work covered. We're good with that. Changes to our prints and mitigations incorporated. We just talked about that. And then we got to be ready with knowledge on hand and it written in. I feel like we'll do good. Like, I feel like if we answer these questions as well as like how we handled the FRR, will be good. You did really good, like reacting to whatever they were doing, like behind the scenes, and then put on the spot. I felt like I did okay, so I'll work on that. And I
You're
think
very
that'll be good.
professional.
Thank you, thank you. I'll stop sharing my screen. But yeah, so that doc um, is on there. We'll go over it in lab today and like add to that.
Yeah.
Any other comments?
So far so good. I definitely think we have work to do, but everyone has work to do, so we'll get through it.
Yeah, we're not alone at least. So okay, I'll stop the recording.